Welcome back to the Deep Dive. Today we're getting into a topic that really defines global mortality, coronary artery disease, CAD. And when we talk about heart disease, we always point to cholesterol, lifestyle, genes, but that whole approach, it misses this huge living system inside us that's driving inflammation.
It really does. So for everyone listening, if you want to be truly informed on cardiovascular risk, you have to look beyond those traditional factors. The gut microbiome has emerged as, well, the pivotal player. It's deeply influencing inflammation, metabolism, and the entire process of atherosclerosis, the hardening of your arteries.
Your gut-heart axis.
In its most dangerous form, yes.
And the study we're diving into today, it takes this to a level of detail we just don't see very often. They aren't just looking at general categories of bacteria. They use these next-gen tools, shotgun metagenomics, and something called metagenome-assembled genomes, or MAGs.
It's an absolute information tsunami, but, in a good way. For those unfamiliar, think of it like this. Traditional sequencing can tell you who's in the room, which bacteria. But MAGs, that's like reconstructing the entire operating manual for specific strains of those bacteria. We can actually see what genes they have, what they're capable of doing.
Okay, that's a critical difference. So our mission today is to uncover these precise microbial signatures, the metabolic pathways, and the functional shifts that separate CAD patients from healthy people. We're looking at a cohort of 14 patients matched against 28 healthy controls. It's about finding the exact mechanism of failure. All right, let's start with the WHO, the big picture survey. The researchers found 15 bacterial species that were significantly different. So who are the critical casualties? What are we seeing vanish in CAD patients?
The pattern of loss is just a classic sign of dysbiosis. The main thing is the loss of these heavy-hitting protective species, especially the ones that produce a lot of short-chain fatty acids, SCFAs.
Right, like butyrate.
Yeah.
And those are key for the gut barrier, for keeping inflammation down body-wide.
Exactly. If you lose those SCFA producers, your gut lining gets weaker. It gets more permeable. And then all these inflammatory molecules can leak out into your bloodstream.
So is there a poster child for this? One species that really stands out?
Undeniably, it's Faecalibacterium prausnitzii. Its reduction is a huge red flag. It suggests a loss of butyrate, which leads directly to that increased permeability, and then boom, higher systemic inflammation. That's a key driver of heart disease.
And I'm guessing it wasn't the only beneficial one that took a hit.
Oh, far from it. We also saw a big drop in Lachnospiraceae isoflavonoconvertins. This one's fascinating. It converts compounds in your diet isoflavones into something called equal. And equal has these potent anti-inflammatory and cardioprotective effects. So when you lose Lachnospiraceae, you're losing a nutritional defense mechanism.
Wow.
Yeah, and then there were others like Coronary and Holdmanella, more SCFA producers. The pattern is just, it's undeniable.
And what about Prevotella-coprae? I saw that was on the list too.
Yes, recently renamed Faecalibacterium. It was also way down in CAD patients, which lines up perfectly with other studies on atherosclerosis.
Okay, so that's who's missing. Now, who moved in to fill that power vacuum?
This is where we see the pro-inflammatory shift. There was an enrichment of several members of the Lachnospiraceae family, specifically one called CAG 303, Sp000437755.
Wait, hold on. That complicates things. Because Lachnospiraceae, aren't they usually considered the good guys? SCFA producers themselves?
That is a fantastic question, and it really gets at the limits of these broad classifications. You're right, the family as a whole is generally beneficial. But the specific strains that were enriched here, like that CAG 303, they've been linked in other work to a higher potential for producing something called TMAO, trimethylamine N-oxide.
And TMAO is a known bad actor for arteries.
A very well-known pro-atherogenic metabolite. So even within a good family, we're seeing these, let's call them antagonistic strains, taking over.
That's a perfect bridge, because now we have to move from who is there to what they're actually doing, the metabolic rewiring. What were the functional pathways that were really lit up in the CAD group?
So we saw 7 pathways enriched in CAD and 3 depleted. And the enriched ones, they really point towards metabolic stress. The most striking, I think, was the big upregulation of the urea cycle and L-citrulline biosynthesis.
Okay, the urea cycle. Help me out here, because I associate that with the liver, detoxifying ammonia.
Here.
Why is that a red flag in the gut for heart disease?
So its job is converting toxic ammonia to urea. In the gut, you get high ammonia when bacteria are breaking down a lot of protein. But the link to CVD is that changes in this cycle mess with arginine and nitric oxide metabolism, and that's been tied directly to endothelial dysfunction, the damaging of your blood vessel walls. High urea cycle activity in the gut is a major stress signal.
And do we know who's driving that? Which
hmm
bacteria?
The study points to the genera Allostipes and Coprococcus as the key drivers. And beyond that, we saw this enrichment in glycolysis III. This was mostly being run by opportunistic pathogens, things like E. coli and K. pneumonia. They're basically burning through glucose really fast to produce inflammatory molecules, just adding fuel to the fire.
So inflammatory pathways are turned way up. What about the protective ones, the ones we need for healthy metabolism?
Predictably, they were depleted. We saw a big loss in L-isoleucine biosynthesis and the whole super pathway for branched chain amino acid, or BCAA, biosynthesis. This lines up so well with clinical findings that link messed up BCAA metabolism to metabolic stress, insulin resistance, and CAD itself. The machinery for healthy energy balance is just being dismantled.
Now, what about the degradation of other things? I saw a note about amino acids and simple sugars.
Yes, the CAD microbiome had a higher potential to quickly degrade things like arginine, aspartate, lactose. When bacteria burn through those simple things really fast, the byproducts can make the gut barrier even more dysfunctional. It's a vicious cycle of inflammation.
This brings us to the really cutting-edge part of this study, the strain-level analysis using MAGs. You described it as having the factory blueprint for a car, not just knowing the make. Why was going that deep so important here?
It's everything. Because community level analysis, it just averages everything out. It misses the critical differences between two strains of the very same species. By reconstructing these microbial genomes, the MAGs, they found these hyperspecific functional signatures that are just screaming inflammatory dysbiosis.
So what did the CAD-derived strains have that the control ones didn't?
Their metabolic weight scores were way higher for two functions, N2 fixation and sulfite reduction. N2 fixation means they're self-sufficient for nitrogen, which suggests a more robust, opportunistic community.
And sulfite reduction. That sounds like an alarm bell.
It should. Increased sulfite reduction can lead to more hydrogen sulfide. Now, in small amounts, that gas can be beneficial. But at high levels, it's tied to chronic inflammation and major cardiovascular dysfunction. It's a classic case of a good thing turning toxic. And on the flip side, the control MAGs were better at aromatic degradation. That's key for processing those healthy polyphenols you get from fruits and tea.
Wait, so this means even the species we think of as good guys could be changing sides depending on the environment. That totally upends the simple probiotic A is always a good idea.
It's the profound nuance that MAGs give us. Take Akkermansia mucinifera. Famous protector, right? Well, the strains from the healthy controls had modules for breaking down complex carbs. Great for the gut lining. Now contrast that with Megamonas fumiformis, another generally good one. The strain from the CAD patients was carrying modules for things like methanogenesis, mercury reduction, and, crucially, lipid metabolism. It suggests a functional shift toward being pro-atherogenic.
It's like functional betrayal at the strain level.
That's a perfect way to put it.
Okay, so this brings us back to the TMAO paradox you mentioned. You said the overall abundance of TMA-producing genes wasn't different. How did the strain analysis solve that mystery?
This is the real aha moment of the study. When they looked at the MAGs, they found a critical genetic detail, a non-TMA producing gene called MTXB. And this gene was found in F. prausnitzii and P. plebeius-A strains, but only in the healthy control group.
So even though both groups had bacteria that could make TMA, the protected strains in the healthy people carried a genetic shield to stop it.
Precisely. The protective nature of these species in healthy people isn't just about making scFAs. It also depends on them being a specific strain that has this MTXB gene, which blocks that final step of TMA production. This specific protective mechanism is just gone in the CAD patients.
Wow. That's a level of precision we can never get otherwise. So what does all this mean for diagnosis? Let's look at the predicted metabolites. What was different?
3 stood out. Inosine was way up in the CAD patients. It's a nucleoside. And while it's linked to vasodilation, elevated levels are strongly associated with existing CAD. And then 2 were depleted in CAD, C18.0E, MAG, and alpha mercalate. Their exact protective roles are still being figured out, but their absence is a clear signal.
Okay, so the ultimate test. Can you use this information to actually tell a CAD patient from a healthy person?
Right.
What's the predictive power?
It's incredibly powerful. A random forest model that combined both the bacteria and the metabolites achieved a mean area under the curve, an AUC of 0.89.
An AUC of 0.89 is That's really strong. I mean, for everyone listening, that means this non-invasive panel, just based on your gut, is performing almost as well as some traditional invasive cardiac screening tools.
Exactly. And the top predictors were a mix of both. The metabolite inosine, the bacterium F. prausnitzii, alpha moleculet. It shows that integrating these features gives you the most complete picture. It could be a fantastic tool for risk stratification long before symptoms ever show up.
So if we tie all these threads together, it's like a double whammy in CAD. You lose your key protectors and their barrier functions, and the ones that are left shift their goals toward creating inflammation through things like the urea cycle.
That's it, exactly. The conclusion is that CAD is marked by this very specific gut dysbiosis. Loss of SCFA producers, enrichment of these antagonistic Lachnospiraceae strains, and these major shifts in core metabolism.
And the importance of those strain-level findings, it just can't be overstated. It's not enough to know you have F. prausnitzii. You need to know if you have this strain that carries that protective MTXB shield. So here's the final thought. Given that we now see good bacteria like Akkermansia megamonas can shift their function toward inflammation in CAD patients, how might future medicine move beyond just simple probiotics? Could we get to a point where we identify, isolate, and then actually introduce specific protective microbial strains, the ones carrying that MTXB gene, for example, as a targeted treatment to prevent or even reverse cardiovascular disease? That, I think, is the frontier we're heading toward.